terminology. If you're not familiar with that, you should know. I may not ask it per se, but I'll use it next. Yeah.
The eicosanoid pathway, is that prostaglandin?
Prostaglandin, cyclooxygenase, leukotrienes. Yeah. So I may not ask those specific words. I may not say, give me the definition of. But if I start using the terminology and then you don't understand what it is, and you come and ask me, well, what does this mean? I will tell you.
So in that pathway, the final two things were prostaglandin and aldosterone and leukotrienes. Those are eicosanoids, or
Yes. Yes. Those are all eicosanoids. That's why it's called the eicosanoid pathway. And some are good and some are bad. And some are, it depends on how, a lot of it's perspective, right? In the amounts. And so this pathway is neither good nor bad. So another interesting tidbit about it is the omega-9s that come from avocados and olive oil will not affect this inflammatory / anti-inflammatory pathway. They don't play into this. So the more of it you take or the less of it you take, it doesn't really affect the inflammation directly through this pathway. Whereas most of the omega-3s and omega-6s that you eat do up and down regulate this pathway, so you can increase and decrease inflammation. All right. We're starting the nervous system, right? And this is actually in chapter 4 as far as lectures. And I embedded it in chapter 4. We're coming back, and then we'll go up to the nervous system directly. So we're going to talk about membrane. Yeah, Aaron. As a Harry-Carey answer? Well, we could make a Harry-Carey answer. What's a Harry-Carey answer? What would it be? Okay. Well, we can use Harry-Carey or Yogi Berra. How about that? So if you're a Cubs fan versus an Yankees. Okay. If somebody down regulates the lights, yeah, the nervous system is chapter 6, but this I actually have in chapter 4 lecture. So if you're following along, membrane potential. So this feeds into all of that. So membrane potential, remember, you are a combination of electrical and chemical reactions. You're not just one. So what is membrane potential? It's the difference in charge across the membranes. Right? It's the difference in charge. So here's a diagram of it. So you have fixed anions. So what that means is that the inside of the cell, when you're talking about a neuron, this also applies to muscle cells, is relatively speaking negatively charged compared to the outside. So when you're measuring the membrane potential, you're saying that the negative is on the inside, right? And it's the difference in charges. That list that you have of extracellular and intracellular fluid ion concentrations is why this happens. There's so much excessive positively charged ions on the outside, and most of that's because of sodium. It is so high and so much in excess that relative to it, the inside of these neurons is negative. Now, there are also some anions, some negatively charged compounds that are always inside the cell, and that also confers this characteristic. They're trapped. They can't get out. Okay, so lots of charge. What's that? A lot of the proteins that make the cell function have a negative charge on them, and they're not going in the organelles, right? They're never going to move in or out. And if they do, they've probably been destroyed and they're being replaced. So it really is a net. It's no net gain or net loss. Then you have other cations like potassium, right, that are attracted into the cell. Remember that inside the cell, intracellular concentration of potassium is much higher than outside the cell, right? It's roughly 140 millimolar or milliequivalents, and then about 14 on the outside. And sodium is not permeable and is actively transported out. So these are the reasons, or some of the reasons, why you get this difference in charge. But the most important thing is that inside of the cell is more negative than outside of the cell. That you have to understand. So when I say where are we measuring the membrane potential, it's the inside of the cell that we care about, or that difference. So you can actually put a voltmeter on it. There are millivolt meters that you could put on a cell, okay, and you can actually measure this. It's no different than reading the voltage coming out of the socket here. Same thing, you put a voltmeter on it, it's probably 110, 120, 115 volts. Your body's working on millivolts. Okay, the equilibrium potential. This gets real quickly into some stuff that people I know get uncomfortable with, so I'm just going to have to spend some time with this. It describes the voltage across a cell membrane if only one ion could diffuse. Right off the bat, is this a real phenomenon? No, this is theoretical. Is it realistic that only one ion would ever diffuse across a membrane? You can't do it. You're a very complex being, right? So this is theoretical. Can you do this in a lab setting in an experiment? Yes, that's where they figured this out. But conceptually, it's very important to understand what would happen if that was the case. It's not real, it's artificial. That's what the equilibrium potential. The equilibrium potential is also known as the Nernst potential. I'll spell it for you. The Nernst potential. They mean the same thing. So this is a voltmeter. So if we took a mini voltmeter here and we put one electrode on the inside and one electrode on the outside, basically what this measures and what all voltmeters measure is how big the hill is for the current to come down. Right? And so it's negative. Now, for neurons, you measure somewhere between 60 and 90 negative millivolts. That's what the resting membrane potentials are, these potentials. Okay, what would determine that? Okay, so here's where the nitty-gritty comes down. If the membrane was only permeable to potassium, meaning that only potassium could leave or enter the cell, artificial, you can do this in a lab. You can make a micelle. You can make a semi-permeable membrane if only this happens, right? It would diffuse until it reaches big E, small k. So the equilibrium potential, or the Nernst potential for K, the potassium. Okay? Potassium is attracted inside the cell by trapped ions, but also driven out by its concentration gradient. So there's an electrical component and a gradient component. Right? So potassium wants to be inside the cell because it's a positive ion, and the inside of the cell has a lot more negatives. Negatives and positives attract one another. Potassium wants to leave the cell because the concentration gradient pushes it out. So what you could do, if you think about this in your head, is if you were looking at a cell, you would see a vector, right, of potassium out, and then a vector to potassium in, and ultimately what you would get is that potassium wants to go where? Out. But it's not as strongly as it would be if these negative ions didn't exist. So here's the electrical portion, and here's the chemical portion. That's why I talk about the electrochemical gradient. You're constantly balancing these two issues, these two forces, the electrical and then the chemical. At potassium's equilibrium or Nernst potential, the electrical and the diffusion forces are equal and opposite. Okay? So when I put this mini voltmeter on a cell that I created, where only potassium can move in and out, they're equal and opposite and equal and opposite is at negative 90 millivolts, give or take. So the equilibrium potential, in its truest sense, is the balance point between the concentration gradient of potassium, which runs out. You have to know that, and you know when you memorize these numbers that it goes from out into out, and the electrical gradient, which goes from out to in, and then this is the net vector. Okay? And that happens, this here happens at roughly negative 90 millivolts. Now, you can calculate it ends up being not that high, but we're just going to use 90 for the moment. At negative 90 millivolts, there's an equilibrium. That's the equilibrium point. Do you want to get to an equilibrium point when you're talking about physiology in a living organism? No. You're dead. Okay? That's why you can't do this in a human. You can't do it in an animal because you have to kill them to do it. Right? You do it in a cell. Okay? So the equilibrium potential, or the Nernst potential, is the point at where one ion has a voltage, right? And the forces are balanced, right? The in and the out. So the equilibrium electrical and diffusion forces are equal and opposite. So this is what actually happens in your body. What ultimately would happen in order to have equilibrium is you would have something like this. This would be the equilibrium potential. This is actual. Does that make sense? So this is what it actually looks like in your body. This is what would happen at the equilibrium potential. So this would be the electrical down here, right? And this would be the chemical up here. And they would be equal and opposite. Is that real? No, this doesn't happen in real life. But it would if you put these artificial circumstances together. This is what it actually looks like. The number is the same. This negative 90. It's still negative 90 millivolts. Yeah, Matt.
Where's the net in the actual?
Where?
So the net in the equilibrium potential is 0, right?
Yeah.
Or there's no movement.
Correct.
So what is the actual movement? Or is it the same thing, just a little...
So if I created a cell that was only permeable to potassium, right? And all of the other forces were still there, the electrical and the chemical, eventually they would equal each other, right? And it would be at negative 90 millivolts. This is what it actually looks like, because it's not, you don't have a cell that's just permeable to potassium. So this is what happens in a real cell, and you do have a gradient that goes out.
Okay.
And that always is like that. That's maintained partially by the sodium-potassium ATPase. This is theoretical, this is actual. Okay? It doesn't ever look like this. Why is this super important? We would have never known what the contribution of the potassium ion is to the membrane potential without separating potassium from all of the other ions. So somebody had to do this experiment where they created a system and they only made it permeable to potassium, and they saw what millivoltage the equilibrium occurred, and they say, okay, that's the contribution of potassium. So take this one step further. Okay, now they did potassium. You're a scientist. You're interested in resting membrane potential of a cell, a neuron. You did it for potassium. Now what are you going to do? Sodium. Then what are you going to do?
Chlorine.
Chlorine. All of them. That's what they did. You can figure out the potential to the resting membrane potential of all of the ions by doing these individual cells. Now, there's a big equation which you have to memorize. It's called the Goldman-Hodgkin-Katz equilibrium equation. And that is the equation for the actual resting membrane potential. It takes all of the ions together. And that's how you can calculate the resting membrane potential. So, if I ask you this, if I want you to calculate the resting membrane potential of a cell, is knowing and I'm going to give you an equation for this, the Nernst potential alone enough? If I want to know what's actually going on, can I just use this theoretical situation? Can't. Okay? Can't do it. But does this tell you, if I had a bunch of these individually, and you had to ballpark, okay, which ones are having the greatest effect, could you do it if you had all these numbers, these millivoltage readings? Yeah, would you be correct? Probably in the direction, but not in the magnitude. Does that make sense? So they did this as theoretical, but it's a really important theoretical. I'm going to make you calculate this on an exam somehow. It's going to come in there. All right. All right, so membrane potential, this is just another look at it. So if you took a swatch in a rectangular form and you looked at negative and positive charges, what you would see is there are more negatives in here than there are here. More positives out here than there are negatives in here. That's all I'm saying. Okay, this is the equation for the Nernst. You got to memorize it. Okay? Or the equilibrium potential. They're the same thing. This is the baby version. It's okay. This is intro phys class. Right? And we took out the ln, we took out r, we took out a bunch of other factors which we're just assuming are constant here. And so when you do that, it's 61 divided by the valence of the element. So if it was +2 for calcium, right, -1 for chloride, you take the log of the concentration of that outside and inside. Concentration. Where are you going to get that from? You memorized it. Shocking. I'm not going to give it to you. Yeah.
Where does the 61 come from?
So the 61 is if you take the temperature and a bunch of other physical / chemical constants, that they add up to 61. So you can look it up online. You can Wikipedia this and see what the actual Nernst looks like. It's slightly more complicated than this.
It's okay.
I don't care. This is the one I want you to know. Yeah, Leonard.
So again, two ion calcium and sodium, for example. Since calcium on the sheet is in molar and sodium is in daily equivalents, can you still compare those two values to since they're different units?
Yeah, you can still do it. Yeah, you should be able to do it either in milli equivalents or millimolar. But if you're not, can you compare them to one another?
Yeah.
Yeah. Well, you need to put them in different. You'd have to convert them.
Okay.
Yeah. So if you want to compare each other, they have to convert. Yeah. Teresa, you have a question?
So EX is the voltage inside the cell?
EX, yes, is the voltage that would be inside the cell if potassium, let's assume that potassium was in equilibrium. It's the equilibrium potential. It's not the actual resting membrane potential of the cell. So resting membrane potential and equilibrium / Nernst potential are not the same thing. They're different. Resting membrane potential is real. Nernst potential is some theoretical value. Next.
What is the balance again?
The valence is the charge on the ion. +1, +2, +3, -1. So each ion. Sodium would be +1 valence. A little bit of valence. All right. Notice that these numbers again, right? Don't worry about these numbers. They're off from mine. I know they are, because every book that you'll ever read has them slightly different. But you're going to use mine, not this. But the point here is, if you wanted to look, and you can calculate potassium, you can calculate sodium, what you'd have to know, and they've done everything in molar here, so you can do everything in molarity, right? 145 outside and 12, I think. Yours are 140 and 12. And yours are 140 and 4, right? And yours are 110 and 10. And then calcium is roughly the same. So it's plug and chug. This is not hard at all. You got to memorize the equation, and you got to know what valence means and what X in and X out means, and then you put them in there. And as long as you know how to use your calculator, no cell phones. You got to bring a calculator. That's my new rule from now on. No phones. All the phones come up. I want to see them all up here. We'll take an example. All right. So bring a calculator. All right. So if you do that for potassium, you come out with an equilibrium potential of -90, and sodium comes out to be +66. Right? Again, if you just look at these two, you're not going to have a really good indication of what the true resting membrane potential of the cell is. It's because calcium's there, chloride's there, magnesium's there, there's iron in the mix, there's bicarbonate, which has a negative charge on it. So you'd have to take them all into account. The resting membrane potential, also abbreviated RMP, is the membrane voltage of a cell not producing an impulse. Again, is the resting membrane potential the same as the Nernst/equilibrium potential? And a resounding no. Good. Somewhere between -65 and -85 millivolts, and it depends on the inside and outside concentrations, on the permeability of each ion. So not only the concentrations, but how permeable an ion is to the cell membrane, which means how likely the channel or transporters that move it are likely to be open or easily open. The resting membrane potential of muscle cells, okay, and neurons is primarily determined by potassium. The concentration and the electrical component of potassium is the biggest determining factor in resting membrane potential. That's why, and you know that because what is the Nernst potential for potassium? -90. And what is the resting membrane potential of most muscle and neurons? Yeah, right here. What if it was sodium? If sodium was the biggest factor, right? If sodium was the biggest factor in determining the resting membrane potential, what would these numbers be? Yeah, that's right. So they're both ends of the spectrum. And these are a lot closer to potassium Nernst potential than they are to sodium. Not even close to the sodium. Therefore, potassium has to be the most important ion in determining the resting membrane potential. Questions? Okay. Again, you can take a look at this, and here you can actually measure that the resting membrane potential here is -70. Okay. Let's throw another monkey wrench into the mix. Remember that the sodium-potassium pumps, or ATPase, is not only affecting the concentration gradient of both sodium and potassium, but it's also what's called electrogenic, which means that just by its functioning, it is going to have an electrical effect. It's going to change the millivoltage. And that's a -3. Whatever the resting membrane potential is that you measure with a voltmeter, let's say it's -70. If it were not for the sodium-potassium ATPase, it would actually be -67. Right? It drops at about -3 millivolts just by its functioning. So it is both electrogenic and deals with concentration. That's why when you give me something about radiance or electrical, I always say give me the electrochemical gradient, because oftentimes when something moves in molecules, it affects both. So the sodium-potassium ATPase is electrogenic. Electrogenic, by definition, means that it has an effect on the resting membrane potential. That's what electrogenic means. conditions, not just the concentration. And it makes sense, right? Because you're moving 3 sodiums out for every 2 potassium in. That's going to cause the inside of the cell to be more negative. And it's by -3. All right, here's another beauty. This is called the Goldman-Hodgkin-Katz equation. You got to memorize it. Yeah. This one's got, so remember, Alexis, you were asking me about what did we take out of the Nernst?
Yeah.
RT and Faraday's constant. And then instead of doing it in logs, we're doing the ln. So if you don't know how to use those functions, learn how to use them. All right. This actually isn't as bad as it looks. I'm going to explain to you why. It really isn't. It really isn't. Okay. Let's take them one by one. P stands for permeability. Okay? So this says that in order to figure out, and Vm is the resting membrane potential, not Ex, right? If I want to find out the resting membrane potential of a cell, what do I need to know? Well, I need to know the permeability of that cell with potassium. That's a number. I would have to provide it to you. Okay? If I wanted you to calculate it. Then I would have to know the concentration of potassium, and O stands for outside the cell, extracellular fluid. I would then divide it by the permeability of potassium multiplied by the concentration of potassium inside the cell. So these you know. These I would have to give you.
All of
The P's, you don't know. I've never given you P's. Right? I would have to tell you what the permeability is to each of those cells. Okay? What is this in actuality? Strip this off. What is this? It's the Nernst equation. This is exactly the Nernst equation. The 61 is RT over F. And instead of doing it in logs, we're doing it in ln. So what? Doesn't matter. Same thing. Okay, that's the Nernst equation. Yep.
When we memorize this, we put 61 there instead of RT over F. We just put 61 in there. What part? Will the RT over F ever change from 61? That's what I meant.
Yeah, no, you couldn't for this one, but you could for that one. But, yeah, you can. For a different reason. Okay, so now you go on to the next one. Now you would do the permeability of sodium, concentration of sodium outside, the permeability of sodium, and the concentration of sodium inside. What is this? Strip this off, strip this off. The rest of the equation is then the Nernst. So all you're doing is taking the individual Nernst equations and you're adding them together. Now, then you've got chloride. Now chloride is, notice the difference. Why is it reversed? Because it's charged. Okay? Why are we only using these 3? These are the major ions, the cations, the electrolytes. These are the 3 in highest concentration. If you know the inside and the outside, extracellular, intracellular, each of these, and you plug it into this equation, the rest of it is at the margins. You're going to get a pretty darn close calculation for what the actual resting membrane potential is. Could you add in magnesium and bicarbonate and all those other ones? Sure you could. You would just do the Nernst equation for all of those, and you just keep adding them. I'm likely not going to ask you to calculate this calculation, right? But you need to memorize it. And you need to understand what it is. Does anybody not understand? So the memorization is easy. Just memorize it. Does anybody not understand what this does? That's important, because I probably would ask you. Give me the Nernst, give me the Goldman-Hodgkin-Katz equation, and tell me what it is. And if you can't explain to me what it is now, and you can't explain to me what it is then you're going to lose points. That's a better option than having you calculate. Yeah. Sure. That's what I wanted to hear. You just saved a bunch of people's butts in this class. Okay. This equation is to calculate the actual resting membrane potential. The Nernst equation, the simple version, for instance, just that, is to calculate a theoretical value for just one ion. This actually, you can actually calculate a resting membrane potential, for real, not just some theoretical number. And it won't be negative 90. It can't be, right? So in this case, it would be something like, I don't know, negative 70. Thank you. Yes, Lynn.
In this equation, wouldn't you just do it for sodium and potassium? Because of the pump, chlorine size, all of it.
Well, yeah, but then you're not just. So Leonard's question is, wouldn't you just do it for sodium and potassium? No, because the pump isn't the only thing that's affecting the resting membrane potential. This is also a major ion because oftentimes potassium and chloride are connected and sodium and chloride are connected. So you can't leave out, if you leave out chloride, you leave out a major portion of the electrical charge. Yeah. These are all electrolytes. These are the big ones. So if you went into a hospital, right, and somebody took your blood and they measured your electrolytes, what would they measure? They would measure potassium, sodium, chloride, magnesium, and calcium. Those are the big five. And of those five, these are the big three. Yeah, Nate. What's that I'm saying? Faraday's constant. Yeah.
So since these are still the big three, why don't they do calcium?
You could. You could. It just doesn't have that big of an effect on this. So this equation can get actually bigger than this. So you can add, you could add on calcium, you could add on magnesium, you could add on bicarbonate. You can make this thing grow. And you would get a more exact number. Yeah.
So are those the major three because they're the most likely to be transported in and out?
Correct. It's a combination of their concentrations and their permeabilities. The other ones are less permeable. And so they're less likely to be moved, therefore they're less likely to affect the resting membrane potential. That's correct. Because all of these, and we haven't talked about this yet, have leaky versions of their channels. There are what are called leak channels, and we're going to learn about leak channels for these two when we get to the heart. Those other channels, you don't really have calcium leak channels. Yeah, thanks.
So ours is constant.
Yeah.
And that's
Faraday's constant.
what's the constant of
I don't remember. I don't have it off. I won't ask you. I won't ask you to actually give you the number. Unless I give it to you, I'm not going to ask you. So I'm not going to ask you to calculate. If I did and you had all the information, this really isn't that hard, right? But I'm not. You got a lot of other things that you have to do. So, but I want you to know what it is, and I want you to know what it looks like, and I want you to know what it tells us. If that's all you come out of this class with, then I'm okay with that. So the fact that you know that this is Faraday's or temperature, that's okay. You don't need to know. All right. Any other questions? Okay. This is an action potential, right? So let's talk about an action potential. An action potential is essentially a graph of the changes in the resting membrane potential. Okay? So on the y-axis, we always have, at least in a physiological system, millivolts. So this is the potential in millivolts. It's usually from negative 90 to plus 20. On the bottom is usually time in milliseconds. So if you think about how quickly electrical signals are conducted through your body and how fast you can react, right? You really don't want to be doing stuff in seconds. We don't do stuff in seconds until we get to the cardiac portion of this class because the heart works at that level. If you were a hummingbird, right, or an insect. Insects actually have the fastest action potentials ever measured on the planet. They would be much faster than this. Right? So we're dealing with a human system. Okay, so notice that this is the local potential, well, no, not the local potential. This is the resting membrane potential. And when something is on the graph, let's say we actually hook this up, we can actually measure the resting membrane potential of the cell. Okay? When you're at resting membrane potential, what is happening to that cell? So this black line, letter. Nothing. That's why it's called resting. Nothing is happening to that cell. Okay. You have a local potential. Something changes. And the potential starts to become more positive. Okay? That positive upshift is called a depolarization. It is depolarizing. Now, if you get a number of positives coming in or less positives leaving, or a combination of both, and you start to get this upswing, if you stay below this dotted line, what will happen to that cell? Somebody other than Leonard. Yeah?
It won't like actually reach threshold, so it won't actually send an action potential.
It won't send an action potential. Nothing. As long as you're below this line, nothing happens. The closer you get to that line, the more likely is you're going to activate the cell, in this case a neuron, and send the action potential along. If you stay below that line, you can get as close as you want, nothing's going to happen. As soon as you hit threshold, you immediately get a firing. This is the finishing of the depolarizing. Usually this happens in a neuron. It's sodium coming into the cell. When you get to threshold, the channels, the sodium channels on the neuron are set to open at whatever threshold is. So if threshold happens to be negative 45, when you get to a potential difference of negative 45 millivolts, those sodium channels are all going to open. And what is going to happen when all of the sodium channels in a neuron open? What's going to happen? All of the sodium is going to rush in because why? The electrochemical gradient favors it down, and it comes in fast. Okay? So it becomes even more positive inside the cell, right? Inside the cell, it's negative. Yes?
Is there any time does the sodium potassium pump get inhibited?
No. Good question. Is the sodium potassium pump inhibited during that time? No, it's still working. So think about how much sodium must be coming in. Now this is milliseconds, right? That sodium pump is cranking away. No change there. All of these channels open up, sodium floods into the cell. Okay? What happens is, and you don't see it here, when you get to usually about halfway, so if this is negative, I don't know, we'll just call it, if this is negative 60, let's call this negative 40 for argument's sake. Those sodium channels are then designed to close. So about halfway up this depolarization, those sodium channels close, but you have an after effect, right? It keeps going positive. All right, now what starts to happen, as those sodium channels are closing here, and you still have an increase, the potassium channels start to open, and that's what causes the repolarization. Potassium now moves out of the cell. So the channels that are potassium channels are set to go off at a more positive value. They all open. Why does potassium leave the cell? The electrochemical gradient. So in simplest terms, right? Goldman Hodgkin cats can calculate this. The depolarizing is because of sodium coming into the cell. The repolarization is because of potassium leaving the cell. Now what happens? Did this thing come back as it repolarizes to resting membrane potential immediately? No, there's an overshoot. This is either called an after potential or a hyperpolarization. That's the way your body reacts. It is not perfect, right? So when it's trying to correct this, remember we're talking about homeostasis, can it immediately go right back to here? No, there's going to be a little bit of an overshoot. Okay? A little bit of an overshoot. And then what happens, it will correct itself with this after potential, and then it will go right back to the resting membrane potential. So this is a graph of what happens electrically and chemically to a neuron. The same thing happens in a muscle and how it gets fired. So if this was a neuron, what you're talking about here, this was a neuron. I'm going to draw you a picture. Right? Here's your neuron. Here's your axon. Right? And here's myelin. It's a myelinated neuron. This area right here is called the axon hillock. For those of you who just had anatomy with me, you remember the axon hillock is where the action potential gets generated. So what's going on there actually is going on here. And then we're going to talk about how you propagate that. Right? Because you can't just have one firing and that does everything, right? It has to continue to happen all the way down the chain so that the electrical signal will go in that direction. This is just what starts the cascade. It's called an action potential graph. Questions on this? Everybody get the basic concept. Something has to trigger this. Now, every neuron, every muscle is triggered by something different. What triggers this? It depends. Could be a change, could be a current, could be stretch, could be a change in a chemical, could be a ligand. It depends on the neuron. They're not all the same. Okay? So there's a trigger. You have to understand this because when we get to the heart, we're going to start messing the system up because it's like anything else in physiology. Nothing is actually ever the same. It's slightly different. This is the simplest scenario, right? This is the most basic scenario. So sodium in, potassium out. Resting membrane potential threshold. Hyperpolarization, after polarization, back to resting membrane potential. Time in milliseconds, potential in millivolts. All right? This schematic actually explains for some. Yeah, Crystal, you have a question? Yeah, no. So Crystal is asking when you're looking at an EKG. It's not. When we get to the heart, I'm going to show you how an EKG is. It's related to this, but it's not this. This is not an EKG. But this helps generate an EKG. Okay? So we're going to get there when we get to cardio. That's in the next block. But that's a good question, right? Just think it. This diagram here is useful for some people for understanding all that I have just said. Okay? Some people like to think in pictures. It talks about, it's a summary of the resting membrane potential. If everything that I said just went in one ear and out the other, take a look at this diagram and see if it doesn't make a little bit more sense to you about what's going on. Okay? It says the same thing that I have been saying. It has all the major players involved, right? And the resting membrane potential. Here you have the equilibrium potential for sodium, and the equilibrium potential for potassium. Here you get a resting membrane potential. The sodium-potassium electrogenic pump is involved. All right, we're talking about fixed anions, unequal permeabilities, and concentration. So that's a nice nutshell diagram about everything that I've been talking about so far. Now, we're not done. We've got to move on to part of the chapter here. Okay. All right. So, chapter 6. Okay. All right. Everybody good? I just saw it. Need a break? Can you shut your brain off for a second? What's that? Martino Moore. Yeah, Martino Moore. All right. What do you think about Martino Moore? Does he have any good stories for you? Yeah. Yeah, no, I'm trying to think. Usually I prepare one, so I'm caught a little bit off guard here. That's okay. What? Redo. No, I don't want to do that. Let's think what we're going to talk about here. Something sports related. I was watching, so this is not exactly a Martino moment, but I was watching ESPN yesterday and I noticed that it was Michael Jordan's birthday. So he turned 52 years old. And the interesting thing is what they did is they went through and they talked about his 10 biggest highlights in his career. And the one that stands out for me, I guess every kid my age when he was playing is the slam dunk contest. Must have had the poster where he's slam dunking from behind the foul line, and they showed that shot. I mean, every kid I knew had that. The amazing thing is that, you know, he did it, but Wilt Chamberlain, if you know who that is, also one of the greatest of all time. He was a center for the Philadelphia 76ers and the Los Angeles Lakers, could actually dunk from the foul line without a running start. Yeah. It's pretty impressive.
No way.
It is possible. There's pictures of him doing it. So Jordan's doing it with a running start. Wilt Chamberlain could do it from no start.
They have that height.
Yeah, his vertical.
How tall is he?
7 feet tall. He was a center. 7, maybe more. He may have been slightly taller. 7 foot. He was over 7. 6'4". 6'4". Yeah. So it was just something very, very. I'm always amazed by what human beings are capable of doing. And then he was showing some pictures of the guy that just won the slam dunk contest this year. It was pretty impressive. Yeah, it's pretty impressive. It was Jordan-esque. I think it was very Jordan-esque. So in the days when Jordan did it, they actually had superstars perform in this contest. They don't do it anymore. Now it's all the rookies, and they're trying to get more people involved. So I'm always interested in human performance, so what people are capable of doing. But yeah, Wilt Chamberlain is the only man ever to score 100 points in a single game. 100 points. Yeah, Jordan was in his 60s and 70s too, never 100. That's just amazing. 100 points. All right, so hopefully you shut your brains off a little bit. Talk a little bit about basketball. All right. All right, let's keep going so we can get some. We're still trying to catch up here. Okay. So some of this is some basic anatomy just to get you situated again, and we're going to go back into those action potentials some more. Okay, so the nervous system has two major divisions. All right, like I said, most of you have had anatomy either with me or somebody else. So, right, there's the central nervous system. Remember, the brain and the spinal cord, and then you have the peripheral nervous system, which is basically everything else. Right? And the neuron we've been talking about is the basic unit of the central and the peripheral nervous system, along with glia. All right, so this is what they look like. You should be able to draw one of these. Again, going backwards a little bit. You have the soma, the dendrites are receiving information. Here's the initial segment. Here's the axon hillock, and here's the axon. Remember, the axon is usually taking information away from the cell body, and then you have the terminals. So this is what it would look like if you actually saw a neuron. It's very messy, right? Lots of, looks like some kind of scary Halloween image. All right. So a neuron is a nerve cell. A nerve is a set of axons. They're not the same, right? A neuron is a nerve cell. They're amitotic, which means they can no longer divide. So for years and years and years in neuroscience, we thought that you couldn't fix the nervous system once you damaged it. We know that's not true now because of plasticity. So, but they don't divide. Within the central nervous system, clusters of neurons are called nuclei. All right, and that's the neurons. What is that same thing called outside of the central nervous system? Ganglia. Ganglia. Okay. Same thing. Nuclei in the central nervous system, ganglia outside. Neurons are not the most numerous cells in the CNS. Glial cells are, and glial cells are much smaller. Now remember, if you took, again, anatomy with me, I made you memorize. I made you memorize 8, but according to Eva, we weren't supposed to know them. That's going to be an ongoing discussion with us until she graduates. Apparently, I never said that you had to know them. Anyway, let's assume that I did. There are 8 different types of glial cells. I'm okay. It's okay. I'm okay. I'm a big boy. All right. So this is what it would look like. This is really important, I think, because a lot of in the phys that we're talking about, you can really get lost in those schematics and those diagrams of action potentials. And what you should really go back to is, okay, here's a neuron, here's myelin, this is an oligodendrocyte if it's in the central nervous system, right? So, and it is because it's in the nervous system. And then outside of this area, you have things like microglia, right? Different types of glia. And you have astrocytes, which is another type of glia, which can attach to capillaries and attaches to neurons. It anchors them. It allows nutrients to flow from the blood vessel to the astrocyte into the neuron. And this is cerebral spinal fluid. So these are ependymal cells. Right? So ependymal cells, I'll remind you, are located in the ventricles. And ependymal cells are the cells that make cerebral spinal fluid. Okay? So you sort of have to keep that in the background. Yeah, Max?
Where are they considered glia?
Yes. Ependymal cells are also glia. So glia, glia, glia. They're in the lateral ventricles and the third and fourth ventricles. That's where all the CSF is made. Yeah.
So we should know that they make that fluid.
Yeah, I mean you should have a basic idea. These are the highest in number. These make cerebral spinal fluid. These act like microglia act like an immune cell that is living in the central nervous system. Right. What's that? No, the microglia. Oligodendrocytes are also glial cells. They make myelin
In the CNS.
in the CNS, that's correct.
And then if there's Schwann cells.
Schwann cells are in the, that's right, in the peripheral nervous system, they do the same thing. And notice that they wrap around. This is really important because when we get to how this signal gets transmitted, you've got to have this picture clear in your mind, right? Because it starts here and it goes down and around, right? So I think it's always helpful to have a little bit of perspective with the anatomy. I know this is an anatomy course, but you got to understand a little bit of it. For most of you, it's a reminder. Okay. Astrocytes support cells. They control extracellular environments in neurons. Microglia are immune cells. There are lots of what are called resident immune cells in the body. So within your lungs, within your kidneys, there are immune cells that just live there. A microglia is one of those cells that lives in the CNS. Much like a macrophage would be in the lung. Okay. The epidermal cells are involved in the production of CSF, and the oligodendrocytes are responsible for myelin. So you got to know a couple of them. A lot of it's just a reminder. And then the Schwann cells are doing the same thing as an oligodendrocyte, but in the peripheral nervous system. This is what they would look like. If you actually look at this, you know, it looks like a some kind of a Greek pastry, right? Phyllo dough. That's what I think. I'm hungry, I guess. All right. All right. So here we get into more of the nitty-gritty of how you transmit an action potential. All right. This is the soma, the cell body. All right. So the cell, inside the cell of a neuron, it looks just like the cell of any other cell. So if you take a cell and molecular, the neuron is no different. It has all the same parts, right? But it also has transport systems. Okay? It also has transport systems. There's two main transporters here, kinesin and dynin, right? And so kinesin here, it allows whatever is produced in the soma to be transported down the microtubule to the axon terminals. What are those things? This is an example of a what?
It's neurotransmitter.
It's a neurotransmitter. That's how neurotransmitters get to the axon terminal. They get made here, but they get transported here. Now, dynin is exactly the opposite. What is that? The reuptake. So when you have the neurotransmitter that is disassociated, it will be taken back up as collected parts. The raw materials will be sent back to the soma and you can put it together. Acetylcholine has this happen at the neuromuscular junction. All right? So you need to know kinesin and dynin, and that dynin is going towards the soma and kinesin is going away from the soma. Now, is this talking about the action potential? No. This isn't the action potential. But is this involved in neural transmission? Sure. Questions? Yeah. Dynin is a protein, a transport protein, okay, that allows things that are taken up by the axon terminal to get transported back to the soma. Whether that's the pieces of a neurotransmitter that it's doing reuptake for, or something that it's intaking that it needs for an activity going on in the soma. This is the transport system. Yeah?
Is that like endocytosis in the axon terminal?
Yeah. So in this case, at the axon terminal, you have endocytosis occurring. Absolutely. And this would be exocytosis. Correct. Very, very good.
It's not part of the action.
No, this isn't part of the action potential. The action potential can occur and essentially occurs along the same area, but you can think about it occurring above that area. Yeah. Yes, so this is, Eva's saying, is this autocrine response? It could be. So it could be that the neurotransmitter is released and some of it could be taken up by the axon terminal, or it could be taken up by the dendrites. So it is an autocrine response too. So I'm really liking what I'm hearing here. What I'm hearing is that people are starting to put pieces together of things I've already introduced. That's the way you should be thinking. Exocytosis, endocytosis, autocrine. All of those things are really, really important. Okay, I think I'll end here. All right. So, Yogi, maybe Howard, I don't expect Aristotle. Important things that we did today. Resting membrane potential, and the difference between resting membrane potential and NERS. Super. Write it down. Write it down on a piece of paper and see if you can define the difference between the two of them. What are the two equations you're going to memorize? Okay, so GHK, the Goldman-Hodgkin catch, which you don't have to calculate, and the NERS, or the equilibrium, which you will have to calculate. I would say those are the most important things. And understanding that action potential graph. If you don't understand that graph, then again, a lot of the stuff that we're going into now is not going to make sense to you. All right. Have a fantastic day. No, it's just general. Could you do it? No. It's just general. You need to know. You know what I think of? Something like that, because I don't see a lot of those. One thing that is a picture on that. Like more problem, but they didn't have a question. It's hard to see. The plan isn't working. But you don't have to actually calculate it. Yes, ma'am.
I got an email from Patty and I thought it was about experimental but even drafted it, and I got an email from Abigail.
Right, he thought, because he asked, let me just shut this off a second. He asked me, he's like, is Laura still doing research with you? I said, you know, Laura.
Oh, experimental. He wants some sort of like a research plan or like something for experimental because that's what it was supposed to be.
Yeah, no.
And I wasn't expecting anything from him for research
Right.
since it was with you.
Because he's, right, so usually one professor.